with Professor Ed Blumwald, who is visiting us here for a couple of days in the scope of a USAID program we've got with the UC Davis. So a long experience with Ed. I think it's a chance that we have him here. So I'll read, the brief of his bio. So he's a sort of multidisciplinary person. Starting with a BSc in physical chemistry in Argentina. He then became a development engineer and worked with a tire company. But he was a bit disappointed with that field of work, so he sort of went back to school, did a new BSc in soil science in the Hebrew University in Jerusalem, then turned to a master degree again in the same university in Israel on nitrogen fixation, then continued at the University of California, Berkeley for a PhD on bioenergetics. Then he became a postdoc at McGill University on a membrane transporter. Then became a full professor on biophysics at the University of Toronto between 1987 and 2000. And then since 2000, he has been a full professor and a professor chair at University of California, Davis. So Ed,
yep,
it's a chance to have you here. The floor is yours.
Thank you.
Ed moves a lot, so for the people that may connect from outside, you may see the presentation, hear Ed, and may not see Ed sort of moving around in the front, right?
I'm shy, I would say like this. So thank you very much for coming, and I really am enjoying the visit to ICRISAT. It's my first time, and I did about it's a great institute. I think that from the CGIAR institutes, you got the best. It's really very impressive. The work is just impressive. No. Almost, but not. You guys are bigger, really. I really enjoy it. And okay, let's start. And I do not even look at my slides, so I hate reading slides. So you guys can read this. Let's start with the first one. But essentially, maybe we should start with the evolution and the way that plants evolve to cope with stress. When there is a drought, or there is intense heat, what plants do, they try to escape the stress. And the way to escape the stress is just to senesce and start dropping leaves. The plant wants to reduce the area of evapotranspiration. The smaller the area, the more water can the plant save. So our idea was very simple. If there is a problem with that, of course, because you drop leaves, the plant drop leaves, and those leaves are still full of nitrogen, potassium, phosphate, even water. So the question is, can we somehow make the plant hold those leaves and use that material in the sources to feed the sinks? We have to also remember that in agriculture, we don't care about the plants really. We care about sinks. Fruits are sinks. Seeds are sinks. Grain is sinks. The plant can look terrible, but if we can harvest the sink, we are happy. So what we decided is we know that cytokinin inhibits senescence. So what we decided is why not to express a gene encoding for IPT, isopentenyl transferase, which is the bottleneck in the production of cytokinin, and have, when the stress appears, cytokinin will be produced, and senescence, stress-induced senescence, will be halted. Of course, there is a problem here that you have to be very aware. You don't play with hormones, because there is a very tight homeostatic relationship between hormones. If you change one, you are going to change the rest. So we needed somehow a way to control that production of cytokinin, and we came to this strategy. We use a promoter of driving the transcription for a gene called SARC, senescence associated receptor kinase. We do not have the foggiest idea what the gene does in beings. We don't care. What we knew is that this promoter was activated by maturity just before senescence. And then we also saw in the lab that was activated by stress. So we wanted this. But we wanted also to generate a self-regulated system. So what we did is we put the promoter driving IPT, and if there is a stress or senescence, stress-induced senescence, the promoter will be activated, and there will be transcription, and there will be the production of cytokinin, and at that time, what will happen is that senescence will be halted, and if we hold senescence, then there will be no activation of IPT. So what we are doing is a self-activated, self-regulated system. And we wanted little bits of cytokinin. Why? The production of cytokinin to arrest stress-induced senescence is not new. The first paper appeared in 1979. There were just dozens of papers. The only problem is that in general they use constitutive promoters. And when you put too much cytokinin, you are screwing the plant. And in fact, too much cytokinin, there is no auxins. No auxins, there is no roots. And so on, so on. No differentiation, no flowering, late flowering. There was a variety of problems. So we decided to go with this strategy. And then we try in the greenhouse. This is published in 2007. And I have to say that in my lab, we are experts in making things work in the greenhouse. Not necessarily in the field. We are going to discuss that. It's not so easy to translate from the greenhouse to the field. But essentially, those are tobacco plants. They were grown to maturity. We halted irrigation for 2 weeks. We just closed the greenhouse and left. Then we rewater for a week. You can see these plants never recover. These are the transgenic plants. The difference is obvious. It worked, at least in tobacco in the greenhouse. And then we try in rice. This has also been published in a series of papers starting in 2011. And essentially the strategy is very easy. We know, and you guys know very well, that the very critical stages are pre-anthesis, post-anthesis. So what we do is we water. We stop watering during pre-anthesis or post-anthesis, and then we continue watering to maturity, and we collect and we measure yield. So sometimes we grow thousands and thousands of plants. We don't care about the plants for 4 months, don't even look at them until they yield. And then we measure grain yield. We see a difference is good. We didn't see a difference. We wasted our time. But it's important to see the phenotype. And you can see nicely, oops, sorry. Oh, I forgot something there, but you can see here the expression. This has been published. The important thing is this. You can see that the expression, SARC is driving the IPT transcription, 4 days, 3 days, and then comes down. In the summer, in California, summer can be, quite hot, 43, 44. In the summer, it's 3, 4 days. In the winter, it's 6, 7 days, depending on the temperature. But essentially, you can see very nice the activation and the self-regulation. And then you can see the yields. We are getting a penalty because of stress, but comparing to the wild type, the yield is much more significant. To tell you that 5 or 6 years ago, you will go to a farmer in California and you will tell him, hey, I have a variety, and if there is a stress, you're going to take a penalty of 30%, will kill you. After 6 years of terrible droughts, you tell them that it's going to take a penalty of 50%, and it's going to take it. Because really it's very, very difficult. Although we have rains now, our water reservoirs are at 38% capacity. We are in deep doo. No water. So, but essentially working rights. So what we decided, one of the things that we know is that you have to be, as I said, very careful, very, very careful when you play with hormones. Because the problem is that there is a very intensive crosstalk. Intensive. So we know about these interactions. We know oxygen jasmonic, cytokinin, ethylene, and abscisic. They play around a lot. And of course, you have to be very aware that if you apply from the outside, it's totally different if you are endogenously synthesizing the hormone. They have different strategies. So you have to be careful. In order to corroborate that, for example, we ran an experiment a few years ago where we did just arrays, and I will not go through this, but you can see that for, of course, cytokinin, but genes related with ABA or with brassinosteroid, jasmonic, oxy, you can read on that. There is a huge change. This is wild type with water and stress, transgenic with water and stress, and the changes are really, really drastic. Although we are changing only cytokinin. For example, just to show you what happened with brassinosteroid-related genes, you can see all of these are associated with the biosynthesis of brassinosteroids or the signal transduction pathways, and you can see that in the most of the transgenic lines, these guys are going up, but in the wild type, whether it's well water or stress, they're going down. So this is are affected by cytokine. So we decided to go one step further and try to apply a systems biology approach to see what's going on with all this and understand the roles of cytokinin during the stress. And essentially, you know, when you use systems biology, of course, you're going to do your transcriptome and now RNA-seq and all that. It's very cheap, lots of data that you have to analyze. And you are going to do your metabolome, and you are going to do the proteome. But for the youngsters, just to tell you, you have to buy one more machine. It's not so expensive, but it's very important, and it's the mortar and the pestle. Because you have to do biochemistry. Without biochemistry, it's like playing with a little Lego. You put the people, you put the houses, you put the cars, but you don't know in which direction they have to go. And you have to remember that over 80% of the reactions, biochemical reactions in biochemistry, in plant biochemistry, are reversible. So you have to be very careful. And of those, 50% are very close to the activation energy, the delta G. So they can go either way. So you have to be very, very careful how you are going to build up your case in the case of transgenic plants. So just to show you, we published this in Plant Physiology not so long ago, so I'm just going to show you this. Here you have everything. In this case, we are measuring sugar and starch biosynthesis. Essentially, what you have here are amounts, activities, because the proteomics and the metabolomics, also the specific activities and expression. I'm not going to go to that. Essentially, what we are analyzing is a relationship between sugar and starch. We have to remember that, as the title says, what we are trying to understand is a relationship between source and sink. Because essentially what is going to give the plant an advantage during stress is the ability to turn over very fast sources to sinks and vice versa. So just to show you, let me show you this, for example, which is the amount of sucrose or starch in the flag leaves or in the grains, and something which is important, you can see red are the transgenic plants, and you can see very nicely that although we are applying stress and we are getting a penalty, nonetheless the grain contains 30% more of starch. And remember, we grow rice because of the starch. We didn't expect this result. We thought there would be a difference. We didn't know which way. This is a nice difference. But that shows us how much work we have to do to understand yet the system. If you go now to the biochemistry, whoa, we have all of this, which is more complicated than the subway map of Tokyo, and you have everything here. And essentially, if you look at transcription, just to understand this, you have here wild-type well water versus transgenic well water, wild-type stress versus transgenic stress. And I'm going to go very short on this. This was also published in Plant Physiology. But essentially, what we are seeing in the wild-type during water deficit, and pay attention, I am not talking about drought stress. There is no drought tolerance in plants. That doesn't exist. Plants need water. Without water, they don't grow unless you're harvesting cacti. So we talk about water deficit stress. And during water deficit, what we are seeing is essentially a huge inhibition in the ability of the plant to produce ammonia. We measure the activities of GS and GOGA, to which extent they can truly assimilate nitrogen, and those are okay. Activity-wise, there is no problem with those enzymes. Although we have to remember GS and GOGA have very low affinity. Really, they are not very specific, but they are working on both. The problem is there is huge reduction in nitrate reductase and nitrite reductase, and there is not ammonia. So what the wild-type plants do is they are trying to get ammonia somehow, and they do is they go very fast from glutamine, deaminate glutamine to glutamate, producing ammonia to feed the cycle. And of course, we have a huge increase in GDH, producing very, very much 2-oxoglutarate. Red implies increase in the transgenic. In the wild type, all the carbon assimilation is decreased because now it's hampered by the activity of GDH because it's totally regulated by how much glutamate is going to be produced. It's a lot of glutamate because all the glutamine is being used to produce ammonia because they cannot take ammonia. As a consequence of that, what we are finding, by the way, the amino acid transferases are equal, there is equal activity, there is no problem with that. So one of the things that we find, please look at the red arrows, those are all the amino acids. The wild-type plant is full of amino acids, full of amino acids. One will say, wow, it's good, full of amino acids. No, it's bad, because if you have an increase of amino acids, you are not synthesizing protein. And what we get is a massive reduction in protein content. This is at the whole plant. You can see very nicely the decrease during water stress of proteins. And we check protease activities, and we check carbonylation, and explains why we have less protein, but protein is not being produced. And this is one of the main problems that you have essentially in cereals. Your carbon to nitrogen coordination went berserk during drought. There is no ammonia uptake, and there is a huge decrease in the feedback between nitrogen and carbon assimilation. As a consequence of that, we have less protein. And this is what I'm trying to show here. Essentially, this process is really inhibited, and there is a massive use of energy that goes in this recycling for ammonia production that really is taking the energy away for synthesis. So we did some field trials. I have to say that when we did this, I couldn't care less about peanuts, and I have changed my mind now. We are working on peanuts. We are collaborating here with Vincent in pear millet and hopefully with peanuts. But when we did this, peanuts for me was attractive because when you look at the plant, you see carbon. And when you look at the pods, you see nitrogen. So I thought it was a very neat system. So we went with 4 peanuts. with Hongshang in Texas Tech University, and you can see very neatly the difference between wild type and transgenic plants under stress. Very interestingly, and now we understand why, we can see also the difference in pod size and number of seeds. This is a nitrogen, and we can see that in the transgenic plants there is very tight coordination between carbon and nitrogen assimilation. They're working well. You can see few lines here. This was done in the field. We have few lines here that are producing more. Under irrigated conditions, there is no difference. We did the same. So what we are doing now is, well, you know this. I use this example when I talk in universities with students, because students usually take the shortcut. They're going to transform a plant. They use the variety of that species which is easily transformable without thinking in its value. We transform the worst of the peanuts. We cannot even improve them. We cannot put them anywhere. Breeders don't want to see it. So we have to go back now. And we went and we asked for varieties which are commercial. We went to the growers, they gave us varieties, and of course, those are very difficult to transform, but it's very worthy. So we did the transformation. These are T0. And one thing that you can see nice is we are getting already the phenotype at T0. We have bigger pods. So we are now in the T1 to T2, and until we get T3, we cannot do anything. But you can see that if you stop watering, they remain greener, which is a good sign. We don't want to kill this because we're trying to get seeds, so we have to keep the plants. So we did the same with cotton, and you can see again the same story, transgenic lines with more yield. Here is a fiber or number of balls, and you can see that it's higher in the transgenic lines. We published that. We just got the paper coming in Journal of Biotechnology where we went and we transformed corn with the INTA from Argentina. They have incredibly good varieties, and we did this. We could not do it in the field, but we use pots that are like 110 liters or something like that. I mean, huge pots with a lot of soil just in case. But the way of do the stress is each pot receives 2.2 liters weekly. And during stress, what we did is we went to 750 mLs, a third. Oops. Oh, it's my computer. Oh, sure. I was blaming you, but no, it's me. I'm sorry. Oh, I'm sorry for that. I apologize. Let's close this. And let's go here. Sorry. And let's go here. Okay. Sure. Okay. Sorry for that. And you can see we went from 2.2 liters to a third, more or less, and then we went to 10%, 250 mLs during 20 days. This is a lot of deficit. And then we rewater, and I'm going to make it short. You can see here well water conditions. There is really no difference. And you can see here the wild type after the stress and the transgenic after the stress. There's a huge difference in the grain, etc. So this is not the best looking maize, but it's the one that they use for breeding. So now they're going to introgress it and get more lines to go to the field. I think we are getting the field permit very soon. Okay, now when we talk about stress, we have a problem. As I said, we are very good at doing things in the greenhouse. When we go to the field, and it doesn't work very well. The problem is that, you can play around with a little bit of water here, water deficit there, but in the field, hey, if there is no water, it's hot. And if it is hot, there is wind. And sometimes your water quality is not the best. So we are trying to devise protocols that give us a very educated information about the plants before we go to the field. Before we come here and we say, Vincent, please do the experiment. Because it's a lot of work associated with that. So what we are using Brachypodium, which is a good model for cereals. It's also another reason we use Brachypodium distachium and Brachypodium sylvaticum, because we are trying to understand the molecular basis of perenniality. And distachium is an annual, sylvaticum is a perennial, so we are getting some experiments there. And you can see nicely control plants, and you do salinity and heat, and drought and heat, and salinity and drought, and all together, and you can see we can get good phenotypes. So we are testing under these different protocols. It's different if it's sylvaticum or distachium, and that change with plant to plant. So you have species, you have to be very careful, you have to redo it again. But that's what we are doing. And from all the data we have to rise, we establish a pipeline. We have all of these genes in Brachypodium, and in Brachypodium, some in distachium, many in sylvaticum, and we are putting some on rice, and now we are going to put a lot in Ceteria viridis, because it's a great model plant, and we find a new way to transform by floral dip. The paper just got accepted in Plant Journal, so soon it's going to come out and you can use it. And it's very efficient. And you can see some previous experiments. Sometimes, HID1 is one of the genes associated with brassinosteroid synthesis. You see transgenic lines, we get good results. Sometimes we use DW5, also associated with the biosynthesis of brassinosteroids. We are not getting good results. So you have to test. Although we are doing genomics and we are doing the analysis of 25,000 genes at the same time, functionally, we have to do it one by one. There is no magic phenotyping apparatus to do that. But let me tell you something about promoters, how careful you have to be when you choose promoters. An industrial sponsor gave us some money because they want to have water efficient alfalfa. And we try also, because of the result we got with rice, maybe we can get alfalfa with more protein content. So we transform some, and I will show you the results, but essentially this is the way we do it. As you know, the genetics of alfalfa is a nightmare. And besides, you cannot go with seeds in alfalfa because you get the huge depression of your genes immediately. So you have to go by cuttings. So what we do is we prepare clones, we root them, we grow them, and then we start doing our experiments the moment we cut the little cuttings. They're growing there, we cut them all uniformly. At that moment, we start the experiment. Yeah, and we are producing hundreds of cuttings every day just to get these experiments. So one of the things is we use heat that give us good things in Brachypodium under the control of PSARC, and look what happened. We are getting no increase in water use efficiency. Blue is 50% watering, red is 25% watering, and the light blue is 100% watering. This is a wild type. And you can see that although we are getting some increase in yield in the transgenic plants, we are getting no effect on water deficit tolerance. And we do not understand why. But then what we did is we used another gene, HSR, which is a heat shock factor, really, HSR1, But we use it with the PSAC, we got the same results than before. We use it with the constitutive promoter ubiquitin. And now what we are getting is, look at the blue, we are getting a beautiful increase in water use efficiency. With 50% watering, we are getting, 60, 50, 60% more yield of the alfalfa. And well, not with 25, but with 50, we are saving 50% water, and yet we are getting more yield. So how come this is working and not? What we discover is that when we make the cuttings, PSARC is being expressed. So our gene is expressed the moment we do the cuttings with the inducible promoter we chose. So we are seeing no phenotype because of water deficit. We see a phenotype of yield. This is very nice. We are getting increase in yield, but not necessarily an increase in water use efficiency because our promoter is being activated during the preparation of the cuttings. And that only points out that sometimes it's not enough to have an inducible promoter. You have to really know the system you are using very, very well and your protocols, because sometimes that is not enough. So you really have to have a number of promoters to use, and we are really spending now a lot of time getting good promoters that are working for the particular plant that we are using. Now, I'm going to go to a different subject. It's not different, but a little bit different. And do you have any questions till now? Are you awake? Good, good. I apologize. I forgot to do this. So one of the problems that we have today is that there is too much information for us. We're talking about systems biology, and then, we have genomics, and we have transcriptomics, we have proteomics, and we have metabolomics. But if you really start looking, I found that in the last 7 years, we got all of these, and we got all of these. So here is a problem. We get this massive amount of information. How we are going to do? In my lab, we don't do screening. We believe in hypotheses. So what we decided is to start setting up some hypotheses and see what we fish. And we decided, this work was done by 2 postdocs, and Song Hu Wong is now a big shot in China, and Nir Sadeh is another postdoc who's in the lab yet. But we decided to do is, let's have some questions. And I give you this as an example of the power of physiology. I call it. Physiology rules. Why? We are going to do data mining. So you can read, but we set up some questions. We have the Arabidopsis genome, 25,000 genes. And we want to find some gene associated with tolerance, although we are very aware that one gene is not going to do the work alone. These are very complex traits. Maybe some genes help us to discover the network associated with that process. But again, I don't believe in magic genes. They're genes that contribute. And then we start saying questions like, for example, what is the main process that is being always a first target of stress, whether drought, whether soil, whether heat? Photosynthesis. The first thing that happened to plants under stress, they stopped doing photosynthesis. So for example, from 25,000 genes, we went to 4,000. All of the genes, all of the genes associated with photosynthesis, all of them, whether biosynthetic, whether no, blah, 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 blah. Okay, and then we said, how many of those genes are located inside the chloroplast? Well, now we went from 4,000 to 800. And then we start asking more questions. For example, we are very aware we did not want stay green. We wanted only membrane bound. And why? Because what we know, not only that photosynthesis is important, but in the chloroplast, there is 70% of the nitrogen of the plant. And the plant has to use nitrogen and have to break down the chloroplast and get the proteins, the nitrogen. But from those 70%, most of the protein is not located just in the chloroplast. It's located in the thylakoid. Those are the proteins associated with photosystem 1 and photosystem 2. Those are the main rich in nitrogen. And the plant recycles that. So asking these questions, we went and we ended up with around, I don't remember, it was 30 or 50 genes. But then we asked another question, very, very important. We would like to get a gene that is in a family of one. We know that in plants are families of genes. Now, in this case, we want a family of one. And why we want a family of one? Because as you know, sometimes to get a grant, you cannot write just transgenics. You have to write other ways. Like, for example, killing or silencing. Now you can do gene editing, CRISPR-Cas9. So we wanted a family of one that can be easily targeted. And surprise, surprise, we ended up with one gene. We got really lucky here. Of 25,000, we ended up with one. And that we call CV, you will see why, chloroplast vesiculation. This has been published in Plant Cell December last year. And what we found that this protein is really, it's everywhere. In every plant has one. Wheat has 8, only expresses only one. Soy has 2, but expresses only one. And this protein is really not very similar, with the exception of these very conserved domains. Okay? So, we decided to check its expression, and you see, if you go checking during senescence, this is Arabidopsis, remember? Senescence, expression goes up, drought goes up, I should say water deficit. You rewater, expression goes down, darkness goes up, salt up, oxidative stress up, heat not. Apparently it doesn't associate with heat stress. Okay, we can live with that. So a very good behavior, actually, according to what our hypothesis. But then we tried to get knockouts, and there's no knockouts of this plant. They don't exist. We don't know why. We could not find them. We decided to prepare our own. But the problem is, we try to overexpress the protein, and we never got transgenic plants. Never. The plants die. So we decided to overexpress it. We decided to overexpress the gene under a total chemical inducible promoter. And what I'm showing here, actually, we label with GFP, and you can see, in this case, you have a stroma proteins labeled red, nothing else, and this is chlorophyll. And you can see here that when you express with dexamethasone, I think it's 8 hours, I don't see very well, you start seeing the vesicles. You see the GFP somewhere there. This is a protein that we want, and is associated with the chloroplast. But after 18 hours, we see that this thing is moving away from the chloroplast, leaving the chloroplast. Whoa. So we do a lot of trafficking and regulation of protein processing in the lab and other projects. So we decided let's stop vesicular trafficking by inhibiting the VATPase in the trans-Golgi network. And when we do that, we see that the protein gets stuck to the outer of the chloroplast, but doesn't move anymore. So what I'm showing you, I'm showing you that essentially the protein goes to the chloroplast and then comes out and moves in the cell. That's interesting. What we found using proper markers, RAB is a marker for prevacuolar compartments, VAM711 is a marker for vacuoles, and what you can see that essentially the protein goes from the chloroplast to the vacuole via the prevacuolar compartments. Whoa, this is interesting. This is a protein that is being produced somewhere in the ER, goes to the chloroplast, then comes out of the chloroplast and goes to the vacuole. What is it doing? Okay, so what we decided is to check, and essentially to make this very short, you can read the paper or send me an email and send it to you, but essentially we can see that the protein enters the chloroplast and the chloroplast goes poof, breaks down. Not with that noise, but breaks down. And then goes to the thylakoid. And now the thylakoid breaks down. But breaks down forming these vesicles. And these vesicles essentially contain our protein. In this case, we just use gold, immunogold, to see it with an electron microscope. And you see the protein in these vesicles. What's going on? Well, essentially, what's going on is these vesicles are being, this protein gets on the thylakoid, breaking the chloroplast. When it gets there, somehow, and I'm going to talk about its function in a moment, somehow is part of this assembly of the vesicle that contains a photosystem 2 and photosystem 1, I will show you that in a moment, and takes everything to the vacuole where it's going to be processed and nitrogen will be recycled. And I'm going to try to make this short. What is the function of this protein? Listen, I go to a lot of biophysical meetings. I like that, and protein synthesis and things like that. And one of the things that in the medical field is quite accepted, that more than half of the proteins don't have a function. Sorry, don't have a biosynthetic function. What they do, they just stand still and they're scaffolds. They allow the binding of other proteins to form the coatomer complexes that makes things happen. If enzyme A has to be with enzyme B to produce C, they have to get together. And these proteins are scaffolds. What CV is doing is providing a scaffold that penetrates the thylakoid, forms part of the thylakoid. We're trying to study now how those lipids do this. It's a very complicated system. There are 4 proteins involved apparently. And by doing that, can bring interacting with proteins of photosystem 2, proteins of photosystem 1, and so on and so on. Just to show you this, first of all, we have 2 other ways to do this. Autophagy is one of those. And senescence-associated vacuoles is the other one. It's a way that chloroplasts are being recycled. But these are not autophagy. You can see nicely with markers that the vesicles are different. And you can see also that in a mutant that cannot produce autophagosomes, CV is still walking to the vacuole. That's very nice. And the second, we did the same thing with SAVs using one marker, SAG12, and we can see very clearly, I don't have an arrow, but we can see very clearly that is different than SAG12, so it's not associated with these SAVs. Essentially, it's a novel pathway, it's a third pathway for recycling, very importantly, membrane-bound proteins in the thylakoids, essentially reducing complexes, photosystem 2 and photosystem 1, where most of the nitrogen is. And you can see when you overexpress, your plants die. And you can, oops, you cannot see because I'm playing around, but you can see that now we are trying to silence it. Our strategy now was, if this protein is the one that is mediating the turnover of the chloroplast, what if we silence it? What if we silence it? And we start by that. We start trying to find targets to produce stress-tolerant plants. So we find CV, we know the function. What if we silence it? Can we enhance stress tolerance in transgenic plants by silencing CV, by diminishing that turnover of chloroplasts? Well, let's see. So first of all, we tried two ways of silencing, and really we are even doing CRISPR, but you will see why CRISPR is very difficult. In this case, RNAi works, but not always. And we did microRNAs, and if we can get over 15, 17%, sorry, 85, 87, 84% silencing, we get the phenotype. And what is the phenotype? When the phenotype is in, for example, you can see in the 3 lines of silencing that after 54 days, the plants already senesce and they are gone. The ones where CV is silenced, they remain and they continue, they senesce less. You can see here in the dark, they are greener. You put methyl viologen, oxidative stress, and they remain greener. And, come on, you can see here, if you do a salt stress, that goes up to 150 millimolar. With Arabidopsis, you have to use really above 100 millimolar to see any stress at all. You can see that wild type under salt stress are kaput and gone, but you can see that the 3 lines where CV is silenced remain greener and okay and functional. You can see under well water we see no difference. Under water deficit, you can see here the wild type in the yellow square, rectangle, sorry, and you can see nicely that after 13 days those plants are gone. When you rewater, they do not recover, but you can see the 3 lines of silent plants are not bad, and when you rewater, they recover the vigor and they continue growing. You can see that the expression is really drought induced. You can see it goes up, CV, and then when you rewater, comes down again. Now, what it does, I do have time, yeah? 5 minutes more, can I? Okay, I will try to go faster now. Essentially now we wanted to understand the mechanism, and for do that you have to do biochemistry. No way, you cannot run away from the biochemistry, you have to do it. So what we did is, it's very simple, we express and we do a lot of mysterious steps to get the proteins precipitated. We use antibodies, we wash them, we have magnetic beads and a lot of things going on. And definitively what we are doing is, I forget the camera, I'm okay now. So definitively what we are doing is we are precipitating proteins that are interacting naturally in vivo with CV. So we purify them as much as we can, and we go for a mass spec. We do MSMS, SCMSMS, and we identify by proteomics the interactive partners. And you can see nicely, for example, that what we find, of course, we found CV. That's the first thing you have to look, whether you get CV or not, and we are getting the same protein. But then you see we are getting components of photosystem 2. Those are good now. Those are all the membrane-bound components here. That's what CV is binding to and taking. If you do co-expression, you can see nicely their co-localization in the vesicles. For example, NADPH dehydrogenase complex, ZIP20, it's there, and you can see it's very well represented. Also, other parts of the complex, and you can see ZIP20 here, again, co-localized very nicely with CV in the vesicles. You can see it here, and even the proteases that are between the thylakoids, in this case is FTSH1, and again you can see them localized. And one thing which is important, that CV vesicles are not associated with the plastoglobules. The plastoglobules are a way that chloroplasts are always regulating size and things like that, energy, by generating the plastoglobules. Nothing to do with that. And essentially, the proteins which are wrapped in CV vesicles, well, the ones that we found are some stroma proteins, essentially photosystem 2, photosystem 1, NADPH dehydrogenase, innate proteins of the thylakoid, and no plastoglobule proteins. How this is going to work with crops? Here we have a problem. And I know that some of you know about stay green. We have to be very careful here. If you are inhibiting turnover of chloroplast, a source is needed to degrade to feed sinks. If we are going to get grain or fruits or seeds, we need that degradation. So at what time you are going to express CV? In fruits, we have a neat uses for this. Because, for example, for fruit production, you can get a very bad quality, say tomato or any fruit you want, and you go and you express CV in the leaves which are source of that fruit. A week before harvest, you spray those 4 leaves, boom, all the material goes to the fruit, and now you got a much better fruit. And we are doing those experiments, and I think it's going to work. But in crops, it's very difficult. And let me show you an example. We try with rice. Well, let me show you this result first. Look what we got. This is the wild type. You can see that Nir Sadeh, the postdoc doing this, is from Israel. He always goes from right to the left instead of going left to the right. The wild type is here. You can see during stress, here it is totally inhibited. Hey, you know what? In our RNAi, for example, lines, we are getting more than double whatever wild type gives. But look how much it is. It's very, very little. We are getting a stay green effect here. We don't want that. So what in this case, what we did was a terminal stress. We just stop watering, and we let the soil water content to go to 10%, which is almost nothing. Because, you know, you have to remember, in the soil, there is never dryness. There's always some water. The roots are always looking for some water. You have to keep a little bit of humidity in the pot. And if you go at 10%, well, the plant will grow, and I can show you that biomass is going up, but we get no rice, no food, no grain. So we start seeing other ways, and in this case, for example, we are doing drought cycles, water deficit cycles during pre-anthesis, post-anthesis, and grain filling. We do 3 of a week each, and you can see that it's working really. In this case, blue is RNAi, 1. This is, sorry, the color, you cannot see it, but this is a artificial microRNA. It's another RNA, but essentially you can see we have lines that are giving us a very, very good increase with respect to the wild type. And in this case, when it's nitrogen deficient, surprise, surprise, the only one that is working very well is the artificial microRNA. And why? Because with the artificial microRNA, we get much efficient silencing than with RNAi. So there is still some protein doing the work. So essentially, this tells you that for some traits, well, we will have to look maybe for other genes that help the thing can work, but you cannot assume whatever you do with Arabidopsis in other crops. You will have to do it again. You are going to find the conditions at which this works. Of course, we are trying other crops, but we started now, so sometime. So essentially what I wanted to show you is that CV is mediating a very novel pathway for chloroplast degradation that is independent of the two known autophagy OSAVs, and essentially what the protein is doing, enter the chloroplast, breaks the chloroplast, associated with the thylakoid after breaking it, and those vesicles now are going to the vacuole in order to recycle the proteins associated with photosystem 2, 1, and others to use the lots of nitrogen there to feed the cells. Okay, I'm going to stop here. This is the people doing the job. These are the guys that are in the lab right now. We have more, but these are associated with the project. These are people that were in the project, and lucky them, they all find jobs in the last 2-3 years. These are collaborators. I have to mention here Indrajit Tutak, which is here in Hyderabad, in Bioseed. Those are the agencies that funded the work. Thank you very much.
Well, thank you very much, Ed, for that seminar. The floor is open for questions.
I told Vincent, if you don't ask, I go and I ask you, so you have to ask questions.
Thank you for the presentation. And as a breeder, I was particularly impressed to see the differences between the wild type and the transgenic. This is not a question, but an observation on the peanut pods.
Yeah.
Wherein you have shown the wild type and transgenic. The number of kernels per pod is a genetic character of a variety. But in the transgenic, we have seen that there's increase in the number of
Yeah.
kernels. Maybe that may need more investigation to.
Oh, I'm sure. Yeah.
That's one thing. And similarly, on the other slides, again showing the pods. So often when the peanut plants are exposed to drought, we get pops, which mean the pods look full and big, but then when we really open, they will be just having some small, tiny kernels. So it will be more convincing to show the kernels.
Yeah, we did that in the paper. We weigh them and we measure the quality and we show there was no difference and so on. So yeah, that is important. Yeah, we are aware of that. We have seen that also. Yeah, thank you.
G'day, Ed. Thanks for that. A lot of it I didn't understand, but it's great to see that quality of research being done at that level. I call myself a physiologist, but a crop physiologist, not a plant physiologist, biochemist. But so my question is, you talked about senescence. Leaves are largely redundant because as soon as it's not long before you have too many, and their purpose of intercepting light is redundant, and so you can afford to lose leaves. If you stop senescence, you're maintaining your leaves, but that's also having other impacts. So that's the carbohydrates and nutrients that would have been released from those senescing leaves were going to form some other function, like improving your root dynamics or whatever. So when you looked at these plants where you halted senescence, there must have been other consequences on the plants that you would have seen in the senesced plants. Those resources were used somewhere else. So did you explore what the consequence on those other responses would have been?
It's a good question. The roots comes immediately to the answer. What happened with the roots? We don't know. Sometimes we see some effects, but we want to do it quantitatively. So what we are doing is we are putting this luminescence gene now, and we are planning to grow these in boxes with soil, but that we can put them in the dark and see with luminescence the organization of the roots. In grasses, we expect changes. We transform with SARC-IPT, I don't know if Indrajit is here, but it helped us transform desert trees. I'm talking trees. They are now trying to deploy them in the UAE to stop the dunes movement. It's a very difficult problem. And we see some negative effect on roots when you are trying to bring them out of tissue culture, because sometimes you got too much cytokinin and inhibits auxin formation. But if you keep your balance of cytokinin well, auxin remains more or less stable. The problem is when you produce too much. What happened with starch? I don't know. And that has to be associated with whatever dormancy development, development the plant is going on. In grains, in grasses, in rice in particular, and in brachypodium, we see no difference whatsoever in the roots. But of course, now we are talking about flag leaves, where 60% of everything happens in the flag leaves. So it's relative. I really don't know, but I don't see something that goes, ooh. But we put this gene in walnuts. Walnuts is terrible. They use a lot of work. In California, we are full of, nuts, not only people. But we put them in walnuts, and in walnuts, I found differences in the roots. Structurally, thickness, and obviously, they were changing. They were dumping some sugar there very fast. But this is the only thing I know. I would love to know, but we will have to collaborate or something because we are full. But it's a very good observation.
Yeah, very, very nice talk. So my question, rather comment, is about the first part, about the hormones, specifically with IPT. So I was with Pioneer before coming here. So we spent almost 10 years on IPT, and we have to kill the project. So you already mentioned, like there is a lot of crosstalk and fine tuning in the hormones, and so my question is, how would you control that in the field itself? In the greenhouse you can, but in the field, how would you control that?
Well, that is why the choice of your promoter is basic here. The problem is that, and that is the main problem when you play with any of the hormones. If you go to a strong promoter, you put too much, and now you are inhibiting every single other pathway. Auxins is the most clear versus cytokine. So you don't get roots. The problem is the promoter and what promoter to use. And it's a very difficult choice because we took SARC 812 base pairs and we did all the promoter analysis. We published a paper on that. We worked like a year on that. Take away one base pair and already the expression goes down to 60%. One base pair. And there are elements which can be activated by anything. So really, there is no secret formula here. I think that transgenesis and genetic engineering, if you want, together with breeding, when they go together in the right background and taking an advantage of the work done by the breeding practice, can go far because now you have the right background to control the response to environment. But you are expecting from one gene to do whatever you want to do in the field, you're dead. Will never happen. There's a lot of epigenetics going on. There's a lot of variability going on. Here is a choice of the promoter and the right background. Now, in industry, you transform and you do 500 events, 1,000 events. Well, I cannot go more than 15. If not, I have to close the lab. I don't have room, space, people, etc. So there is a lot of things associated with that. But the bottom line is the right line, and you are depending on the breeding practice. You have to talk with the breeders. It's critical. And second, the type of promoter you are going to use. Okay.
I'm particularly interested in knowing if you looked at the pod wall of the transgenic peanut plant. Was that one of the criteria? Because these are drought tolerant transgenic peanut plants, so they're very well known to people. So did you look at some of those traits which could be useful for
Please don't take me wrong. When I did these experiments, I did not know an iota about peanuts. Neither my collaborators so much. Now, I still don't know an iota, but I come to the right place. You know, I really did not look at that. Absolutely. But peanuts for me was fascinating in the sense that you can see carbon, green, nitrogen pods. Just that simple, that simplicity for me was very attractive. But nothing else. Now I'm learning, slowly.
Nice presentation indeed, Ed. Again, I'm back on your IPT strategy. I'm just wondering, in a crop like pearl millet, where almost 80% of the root development takes place within initial 30 days. So in this system, if there is a stress, say around 25 days, so this kind of inducible promoter with feedback loop is going to work or not, especially on the root development.
The root is a bit difficult, but again, I have to ask you, in the first 35 days, how much water you have in your profile, in your soil profile? Because when you put seeds and you start your crops, usually you put water there. There's no just water deficit. There is enough water to give that seed an advantage and start growing. And usually you start drying relatively slowly, and there is also some water there. I will not worry about the roots immediately. I will worry really about photosynthesis first, because you need carbon, you need sugar, and you are going to uptake whatever you can from the soil. You don't uptake sugar from the soil. You get sugar from the top. So as a strategy, I will not worry too much about the expression of Sarc-IPT in the roots, but I will yes do it on the upper part of the plant, because my objective is to try to preserve photosynthesis at any cost. We have a couple of papers in plant physiology in 2009, 10, where we show on these plants a strong effect on ROS homeostasis, reactive oxygen species. And that is what you have to stop. You have to stop oxidative damage, which usually is very, very rapidly advancing during the water deficit. I don't worry too much about the roots now. But again, I don't have Sarc-IPT in sorghum or millet yet. Hopefully we will.
Maybe I'll take a question before Peter want to get another one. But in continuation to that, I mean, you said that in your talk that the first thing that what happens to plants when they're under water stress is photosynthesis. But actually, I mean, when plants are under water stress, they close stomata first. So nothing happens to the photosynthetic systems, close stomata and everything. And then after that, you eventually have damage on the photosystems.
Yeah.
But so how you resolve the two, in the end? Because I mean, there's a part of the water stress where really virtually nothing happening to photosynthesis. So it happens really late when pretty much the crop is over and almost. So how do you, when your 4,000 genes and your 800 genes, I mean, wouldn't it be a better strategy to focus on what really controls stomata?
Our best strategy is, if it's not broken, we don't repair it. And this is important because when you talk with breeders, there are a huge variability in stomatal conductance. And there are varieties which have much more conductance than others. So here is the choice of what you're going to transform. The easy way is to transform something which is easily transformable and is good for nothing. We are going the other way. We are transforming elite. And we are transforming, for example, varieties that are showing in the field have much more control than that. For example, there is gas exchange at very, very low soil water content. Maybe little one, but we don't need too much. So I am escaping the question because I don't know how to deal with that. It's a very difficult question how you deal with ABA. If you close stomates, there is no CO2. Forget it. Oh, you don't lose water, but you don't grow. So you need some of that. The moment you touch some ABA, well, we have a nice paper with Ron Mittler published in Plant Cell this year in 2015 where we show the association between ROS and ABA. So you have to be very careful. You start changing that, reactive oxygen species start chewing your enzymes. So it's a very difficult question. But for that, let's go to the breeders. Remember, I only use Arabidopsis. I am not the Arabidopsis guy. I do not believe in that. But there are varieties that have learned to do that. So I obviate that question going to those varieties.
Thanks for the presentation, sir. And one of the slides you have mentioned 10% of soil moisture.
Yeah.
It doesn't contribute to the plant in terms of grain weight. So whether it is available, the soil moisture stored one is available for the plant, because wilting point, as a wilting point, it differs from soil to soil and depth. So I want to know whether the soil moisture is available for the plant, and after that it doesn't contribute to the field.
Well, we do it by weight. You know, we weight and yeah. So there is some water because the soil is heavier. The root takes it or not, I don't know. I just don't know. But for example, if you do 10 and 0, you see some difference. So some of that water is being used.
Because
Yes.
I have read that in chickpea, for example, though there were 24, wilting point is differed from 21 to 18, but it is not available to the plant, though it is available.
But that
So in
will
that case,
depend.
we can't correlate that in terms
That
of
will depend on your soil.
Yeah.
It's your soil structure. We are using typical soil of the Sacramento Valley, which is very sandy. So water content goes down fast, but it's available. It's not, you know, your red soil that is going to, where your tension pressure is such that can you bring your water out, although it's heavier. So that depends on your soil, really, not on your plant.
Just quickly, I'm picking up on something you said early. I think I quoted you right. Well, I've written it down. There's no drought tolerance in plants, does not exist. Is that what you said?
Yeah,
It's water deficit.
yeah,
It's a response to water deficit.
yes, sir.
I want to support that, and because I see somewhat of a sloppiness in the use of drought in
Absolutely.
this institute and what we write.
I don't know that. I don't know that.
Well, I do. Vincent knows. So I think that's an important point. It's what we're looking at is water deficit, and particularly when we're looking at traits, what we're trying to understand what the mechanism of the trait is and the genes are, rather than just saying it's drought.
Yeah, but you see it in the literature. I'm an editor-in-chief of a journal, and I see drought, boom, I send them back and say rewrite that, because it's misinformation. Plants cannot do it without water, period. Plastic flowers, maybe, but no plants. So unfortunately, we get this drought tolerance. Plants cannot tolerate drought. It's tolerate death. Yeah.
Right. well, let me conclude. I mean, I think it's interesting to have someone, I mean, working with transgenic that say that drought tolerance doesn't exist. I mean, if I say it, I keep on saying it all the time, but people don't believe in me. You know? So to take your quote and make sure you say it in the take two video.
Oh yeah.
So let's give a last round of applause to Ed before the
Thank you.
Wednesday
Thank you
Meridian.
very much. Thank you. Thanks.
Thank you very much.